Hello students, welcome to the new lecture, and in this lecture we are going to start with the observation of lactobacillus bacteria. So in the last class we have stopped here, okay, observation of lactobacillus bacteria. So that is activity 3. So and good afternoon to everyone, and if you have any doubts or questions, post it in the live chat section so that we will discuss, okay, we will discuss about your questions and doubts. So okay, good afternoon everyone. So first is observation. Observation of lactobacillus bacteria. What is lactobacillus? Lactobacillus is nothing but it is a bacteria. So bacteria present in curd. Okay. So it is present in curd or buttermilk. So for this activity, we have to take 2 to 3 spoons of 2 to 3 drops of buttermilk. So take a slide. So 2 to 3 drops of buttermilk on it, and also spread it, okay, and also spread on the slide, okay. So after spreading, okay, what you have to add, we have to let it to dry, okay, expose it to the lamp. So bring near to the lamp. Let us consider this is a lamp, okay. So bring it near to the lamp so that due to the heat the water will get evaporated and it will get fixed. It means the layer of the microorganisms are going to get fixed to the slide, okay. So this is the fixation process. After that we have to add a stain. What is the stain? Crystal violet. We have to add crystal violet. So after adding crystal violet, what will happen? It will get stained, okay. The layer, the smear will get stained. So after staining, after adding the color, so we have to wait for 30 to 60 seconds. After waiting for 30 to 60 seconds, what will happen? So till the time the bacteria will take all kind of color, okay. The color will be taken. So then after 30 to 60 seconds, we have to wash it. We have to wash it with water. So then dry the slide. So then take the slide and observe, okay, observe under microscope. So for the clear observation. And a very good afternoon to everyone, okay. So next is, if you observe under the microscope, you can see such kind of a picture. So this is a picture, or this is not a picture, this is a observation, microscopic observation. So these rod shaped structures are nothing but lactobacillus bacteria, okay. So here, if you see in the bacteria are of different types. In those bacteria, some are symbiotic. What is meant by symbiotic? Symbiotic bacteria means, so this bacteria is closer associationship with the host. For example, in our stomach, in our gut or in our stomach, there is a bacteria. So what this bacteria is doing? This bacteria is going to provide some kind of nutrients. It is going to provide some kind of nutrients to our body. And in return, what we are doing? We are providing food. We are providing food and shelter. Okay, we are providing the food and shelter to the bacteria. It means, what is happening here? Here the bacteria is getting benefited and we are going to get benefited. It means human beings and bacteria both are being benefited. Such kind of process is known as symbiosis. What is symbiosis? Here the host and bacteria both are being benefited. Here, like example, human beings and bacteria both are being benefited. That is known as symbiotic. So others are parasitic. What is parasitic? So parasite means it is also a microorganism. It is going to cause disease to the organism. For example, malaria. Okay, if any person get malaria, what is happening? So that person is going to suffer a lot. Okay, if not treated, that person may die. Okay. It means here the malaria is a parasite. Okay, and malaria is a parasite, and here the bacteria which is present in the gut is a symbiotic microorganism. It is like a friend. So next is the bacteria on the skin. On your skin also there is a bacteria. What this bacteria is going to do? This bacteria is going to help you. Okay, it is going to help your skin. So it is one of the most important things. So symbiosis means the closer associationship where host and microorganism both are being benefited. So parasite means what? Disease causing organism that cause damage to the host. That is known as parasite. So the microorganisms can live in different climatic conditions. What are the climatic conditions? Example, high temperature. Okay, high temperature is one of the climatic conditions. Medium temperature, low temperature. These all are the different types of the climatic conditions. Okay. And by the way, if you have any doubts or questions, post it in the live chat section so that I can read your doubt or questions and I can answer to your doubts or questions. Okay. So next is microorganisms have different sizes. Okay. So one of the largest size is 0.75 millimeters. So its name is Margarita. Okay, Namibiensis. Margarita namibiensis. It is the biggest bacteria. Its size is about 0.75 millimeters. So you can see with your eye, okay, without any microscope, you can see this bacteria clearly. Okay, it is discovered by a person known as H.D.N. Schluids in the coastal water of Namibia. So it is in the Namibia region. Okay, so this is about parasite, symbiosis, and also largest bacteria. Okay. So now our next aim is to observe algae. So what is algae? Algae is a photosynthetic organism. It will perform photosynthesis. What is photosynthesis? Photosynthesis, it is a process of production of own food material. So it means these algae is going to perform photosynthesis. With the help of photosynthesis, it is going to produce its own food material. So this is known as photosynthesis. So example for the photosynthesis in the algae or chara. It is one of the algae. Next is Spirogyra. It is also one of the type of the algae and so on. So next is the algae can be seen with unaided eye. So what is meant by unaided eye? Unaided eye means without any help. Okay, without using without using your microscope, you can observe this algae. So where you can observe? Wherever there is water. Okay, wherever there is water on the rocks, you can observe this algae. Okay, and here next is activity 4. Okay, so in this activity 4 we are going to observe microalgae. So what is meant by microalgae? So microalgae is notably greenish string like structure in water bodies. In the water bodies you can see algae. This algae is greenish string-like structure. Okay, it means it is looking like a thread. Okay, small thread. So if you go to the nearest pond, what you can see? So Amaravati, sir, how to listen previous class of 8th class biology in YouTube. So in the playlist, go to the YouTube. Okay, so go to the channel. Here you can see home. Okay, after home you will see videos. Okay, so after videos you can see the playlist. Okay, playlist. So select the playlist. What is your first chapter name? Your first chapter name is what is science? Okay, what is science? Okay, so this is the first chapter. So you can see the playlist containing what is science. Okay, what is science? Click on it. If you click on the what is science, you can see number of lectures. So number of classes. How many total? 1 to 10 classes are there. So 1 to 10 classes are there. This is the 10th class. Okay, it means 10th lecture. So you can watch all these things. So go to the playlist and select the playlist containing name known as what is science. This is your chapter name, first chapter name. So after the first chapter, second chapter is there. After the second chapter, third chapter is there. That is running at present. Okay. So okay, Amaravati. So you can see. You can now see all lectures from the first chapter to third chapter. All lectures you can see, and also you can write notes also. Okay, you can copy notes. So for each chapter you have to write around six pages should be there. For each chapter 6 pages should be there. That is your notes minimum. So if you feel the notes is lengthy, you can decrease the points. Okay. So this is about not only Amaravati, anyone can attend the lectures again. You can watch the lectures again if you miss, or if you want to watch again, you can watch the lectures again. Okay. So okay, Priyanka, good afternoon. So, okay, Amaravati, I think your doubt is cleared. So now we will go back to the lesson. Okay. So place a few strands. So go to the pond, collect the few strands. Okay. So and place it on the slide. So take a slide, microscopic slide, place it on the slide and keep it under the microscope and observe. If you observe under the microscope, what you can see? You can see these strands. Clearly you can see these strands. So compare your diagram with a textbook diagram. So after seeing the under the microscope, after seeing, compare your observation with a textbook diagram. You can clearly differentiate between those two. So those two diagrams, your observation and also textbook diagram, looks almost similar. So here, if you see, the nearly half of the oxygen on the earth is produced by the microalgae through photosynthesis. Whatever the oxygen that you are getting, you can say that 50%, above 50% of the oxygen is produced by the microalgae only. Okay, most of the oxygen is produced by the microalgae. Okay, next is observing protozoa, that is activity 5. So in this activity 5 we are observing the protozoa. Okay, so what is protozoa? It is a microorganism. So to get a protozoa we have to do something. Okay, we have to do some procedure, that is go to the nearest pond. Okay, go to the nearest pond. So take some kind of hay. Okay, hay is nothing but so rice grains or rice. After taking rice you will get the hay, no, in this way, the strands. Okay, so take those, so and soak it in the pond. Soak the hay on the pond. This is water. Okay. So wait for 3 to 4 days. Come back after 3 to 4 days. Collect water. Collect a few drops of water. Okay. Collect a few drops of the water, and this is known as decoction. So how do you prepare the decoction? Select a pond. In the pond, put hay. Okay, hay in the pond. So this hay is nothing but so they are plant material. So wait for 3 to 4 days, and after 3 to 4 days you will get the concoction, or you will get the decoction. So take the decoction, go to the laboratory, put a 1 or 2 drops. Okay, 1 or 2 drops on the slide. Okay, observe under the microscope. If you observe under the microscope, you can see these structures. You can see these structures like amoeba. You can see amoeba. You can see paramecium. You can see verticella. So these organisms are there in the concoction. These organisms are there in the decoction of the hay. Okay, decoction of the hay. So next one is observing microarthropods. So in the last classes also we have discussed what is meant by arthropod. Arthropod are nothing but minute organism. So minute means very very tiny. Okay. So very very tiny organisms. These tiny organisms are known as arthropods, microarthropods. So these arthropods have jointed legs. For example, here you can see in this diagram, you can see they have tiny tiny legs. In these organisms, they have tiny tiny legs are there. So these tiny legs are known as arthropods. Arthro, the term arthro means, okay, ARTHROPODS. The term arthro means joint. Okay, joint. Pores means all legs. Okay, pores means legs. It means these organisms have the jointed legs. You can see in this diagram, all these organisms have the jointed legs. So some of these organisms are going to cause diseases. Example, scabies. Scabies is a disease caused by the scabies mite. Scabies mite is going to cause a disease known as scabies. It is skin disease. Okay, scabies is nothing but skin disease. You might have seen in some people, so all the time they will itch their skin. Okay, that itching of the skin is due to one of the disease that is known as scabies. Which organism is going to cause disease? That is scabies mite is going to cause the disease known as scabies. Okay. So these organisms are very important in increasing soil fertility. Not all arthropods are dangerous, only some are harmful, remaining all are not harmful. So what they are going to do, they are going to increase the soil fertility, and also they are also found in the bedding, rug, skin, eyelids, etc. So if you see here, this one, okay, this organism is present on your eyelids also. Okay, on your eyelids, this organism is present. Okay, this is known as eyelash mite. Okay, So on your eyelids, these mites are present. So in your bed also, in your bed, in your rug, on your skin, these most of these microorganisms are present. Okay. So soil is rich in microorganisms, especially bacteria, fungi, protozoa, all, and also along with microarthropods. These all are the things that is present in the soil. So if you take 1 acre of land, okay, if you take 1 acre of land and also 8 inches up to the depth of 8 inches, okay, if you take in that up 1 acre of land up to the depth of 8 inches, you can find 5.5 tons. 5.5 tons means a lot of bacteria. So 5.5 tons of bacteria can be found in 1 acre of land, so at the depth of 8.8 inches. So this such kind of huge amount of bacteria or microorganisms is useful for the plant growth. Plant can grow better in these conditions. Okay. So next is excessive usage of the pesticides cause the death of the microorganisms. Some people will use more pesticides in agriculture. So those pesticides will fall on the ground and they are going to cause the damage to these microorganisms. Okay. So observing soil microorganisms. This is activity 6. Here you can take this glass of the water and mix with some soil. Okay, and mix with some soil in it and then stir well. So this is a glass. So it is going to contain some water, add sand and also stir well. So after stirring well, what will happen? Sand, larger particles will get settled at the bottom and small lighter particles will be on the top. So wait for some time, everything will be settled. So after settling of the water in the glass, okay, this is dust particles, so this is the clear water. So take the water here and also put it on a slide, okay, put a drop of water on the slide and observe under the microscope. So if you observe under the microscope, you can see different types of microorganisms. So if you observe under the microscope, you can find different types of microorganisms. Okay, so these all are the different types of microorganisms. So arthropods, protozoa, algae, everything you can see in this in the soil. Okay. So next one is viruses. What is meant by virus? Virus is a different type of unique microorganism. So and by the way, before going into the topic, if you have any doubts or questions, feel free to post in the live chat section so that we will discuss your questions or doubts. Okay. So next one will be the viruses. These viruses can be seen through electron microscope. What is electron microscope? Electron microscope is a highly sophisticated microscope. Okay, highly sophisticated, and it is going to maximize the objects many thousands of times. Okay, so they are living inside the host, and also these viruses are going to live inside the host. Okay, so when the virus is inside, okay, when the virus is inside the host, this is the virus. When the virus is inside the host, it is living. When the virus is outside the host, it is dead. Okay, it is not living. Okay, so this is the reason. So when the virus is inside the cell, it is living. When the virus is outside the cell or outside the host, it is not living. So these viruses are going to cause many diseases in plants and also the animals, and they are going to multiply in number. Example, polio, swine flu, smallpox, these all are the diseases. These diseases are caused by the different types of viruses. Okay. So microorganisms lives as a parasite inside the host. It means when the virus is living inside the cell or inside the host, it is known as parasite. So when the same virus is living outside the host, that is known as free. Okay, that is known as free, means they can live freely outside the host, or they can live inside the host. Okay, but generally when the virus is outside the host, it is not living in condition. Okay, and these viruses can be living in many different types of climatic conditions like cold, hot, marshy land, etc. So these all are the different types of climatic conditions. Marshy means salt or some other kind of water. So these are nothing but about the viruses. So if you see under the electron microscope, you can see such kind of structure. So this is the this is the image of the viruses which is seen under the electron microscope. So next one will be the tobacco mosaic virus. Okay, so what is tobacco mosaic virus? It is a virus that is going to cause disease in the tobacco. So that is, this virus looks in this way. If you observe under the microscope, you can see such kind of a such kind of a virus, such kind of structure can be seen under the electron microscope. So next one will be the bacterial staining. So what is meant by bacterial staining? First of all, what is staining? Staining is nothing but coloring. So coloring of any substance, that is known as staining. So before we go into the topic, and if you have any doubts or questions, feel free to post in the live chat section. So, and I'm able to see no one is asking any doubt or any question. So feel free to post doubts or questions in the comment section. Okay, and remember one thing, you have to write the notes also. Okay, copy these notes. So copy these notes so that it will be more useful in the examination. Okay, so staining is a procedure used to stain the bacteria for observation under the microscope. So first of all, what you have to do, so take a slide. Okay, make a smear. Smear is nothing but a layer. Okay, a layer of the bacteria. So a thin layer or film of the bacteria. So this is slightly heat the slide. If you heat the slide slightly, this layer of the bacteria is going to fix to the slide. So after fixing, what we need to do, we have to add a dye. Okay, what is the dye? That is staining. Example, you can use crystal violet. You can use crystal violet to the smear of the bacteria. So, and wash after 30 to 60 seconds. After 30 to 60 seconds, wash the slide. So, and after that, observe the slide under microscope at 25x. Okay, at 25x or 40x. So if you observe under the 25x or 40x, you can see the clear structure of the bacteria. So Anusri, what is bacterial staining? So bacterial staining is nothing but coloring. Okay, so if you see bacteria is transparent in this way, transparent. If you use the stain, so the bacteria will be having some kind of color. So if it has some kind of color, you can clearly observe under the microscope. So staining is nothing but using some kind of colors to the bacteria, so that it will be clearly visible under the microscope. Okay. So this is about the bacterial staining, and if you have any doubts or questions, ask in the comment in the live chat section. So this is about today's topic, and in the next class we are going to discuss about the chapter 3. So the chapter 3, it is about this second part of the chapter 3. Its name is the world of microorganisms, the part 2. So here we are going to discuss about first introduction. After introduction, we are going to discuss about the topics. Okay, main topics. Okay.